Well, thank you for the introduction and thank you for the chance to speak to everyone today. I'm going to be chatting a little bit about the work I'm doing, somewhat regarding diet microbiome interactions, but in terms of C. diff as a pathogen interacting with a diet that we consume. And I want to start out by a little motivation about how I approach some of the work that I do and what motivates it. Um, and it starts with the idea that our modern diet is incompatible with our evolution history. So if you think about human evolution and break it into phases based on how we acquire the food that we eat, there are three main phases. There's foraging, where we hunt and gather for food. Then there was an agricultural revolution about 10,000 years ago. And most recently, about 300 years ago, we had an industrial revolution where essentially the food industry figured out ways to mass produce certain items or certain nutrients very cheaply. And this changed the nutritional landscape for humans, especially in the Western world, where now we consume a lot less fiber, a lot more salts, fats, additives, and sugars. And what's one important qualification here is that 300 years seems like a long time, but it's actually quite short in terms of human evolution, but not so for microbial evolution, which can adapt very quickly to very rapidly changing environments. So when you think of for example, about certain sugars that we're introducing to our diets that we cannot absorb, microbes can figure out very quick ways to derive or absorb or take up. And we still don't know the consequences of these sort of industrial revolution brought on changes in our diet. But we do have some examples, for example, from the lab of my mentor, Rob Britton. Right before I joined the lab, they published some work showing how a certain sugar may have played a role in the emergence of certain C. difficile rabbit types. And I'll talk a little more about rabbit types and C. diff, but basically there are certain lineages of C. diff that tended or currently dominate in terms of epidemics. But if you look prior to the year 2000, there were only a handful of outbreaks attributed to these lineages, which are rabbit type 027 and rabbit type 078. But around the year 2000, a Japanese company figured out a way to mass produce trehalose, a disaccharide, essentially two sugars linked together, and in a very cheap way, and it has very attractive chemical properties, and it's fairly sweet, so it's been introduced heavily in the food industry. And afterwards, we started to see these global outbreaks of these two rabbit types of C. diff. So a very simple introduction of a sugar that's naturally available in plants or fruits, but now we introduce on much higher levels, has been linked to the emergence of these two particular C. diff rabbit types. And the idea that metabolic traits may play a role in the emergence of a pathogen or specific strains of a pathogen is not necessarily new, and it's not limited to C. diff. But there was also another study for C. diff about a year ago where they did a very large comparative genomic study, and they tried to see what's different about certain phylogenetic clades. And they grouped them into clade A and clade B. Unintentionally, clade A ended up including mostly human-associated rabbit types, but that's basically how these things evolved anyways. And clade B tends to be dominated by animal-associated rabbit types. And when they looked at genes that seem to have positive selection ongoing, and they were associated with clade A, which is the human-associated rabbit types, they included genes involved in metabolism, such as phosphorotransfer resistance, fructose and mannose metabolism, as well as cysteine and methionine metabolism. So it does add another dimension about what could be contributing to the distribution of certain rabbit type of C. diff. Now, stepping back a little bit, what is C. diff? It's a gut pathogen. It's the main cause of hospital-acquired infections in the Western world. And it's a emerging or a major antibiotic threat, as indicated, for example, by the CDC. These are numbers that were fairly recent, showing that there's about 250,000 cases of C. diff annually. And I put an asterisk there because this is only including hospitalized cases or hospital-acquired cases. There's actually as many community-acquired infections as hospital-acquired infections. And what's important here is that typically the main risk factor for C. diff is antibiotic usage in a hospital setting. So somebody's taking antibiotics because they're sick or they're undergoing surgery or chemotherapy, and their gut microbiome is disrupted, and then they get exposed to C. diff spores and they get infected. But there's also a whole slew of community-acquired infections that cannot be traced to hospitalization or to antibiotic usage. So it's still unknown what makes somebody susceptible to C. diff. And it's possible that diet does play a role based on our work and other groups' work. So a bit about the typing and the classification of C. diff. So help us better understand the epidemiology. Typically, or in general, there are 5 consensus clades, clade 1 through 5. And within each clade, we typically group closely related strains into ribotypes. And ribotyping is essentially ribotypes are identified by gel electrophoresis after amplifying for the intergenic space or region between 2 ribosomal RNAs. But basically allows us to group closely related strains and to also track them in terms of space and time. And it helps a lot with understanding how C. diff gets transmitted. But also there are certain ribotypes that have very unique characteristics. For example, ribotype 027 is the most dominant epidemic ribotype globally, and also is one of the more hypervirulent strains. So recently, the Kevin Gary group at the University of Houston, one of our collaborators as well, have been over the past decade tracking the C. diff cases in the state of Texas. So this is a plot that I made based on a table from the recent surveillance study, which is you can find the information on the bottom. Basically, on the y-axis, you have the ribotype frequency in terms of the samples of C. diff they received in the particular year. So for example, we focus in 2018, there's a couple of things I want to highlight. There's only a handful of ribotypes. These are the ones called in blue, yellow, green, and red and purple that make up almost half of the infections that they studied. Most of these infections can be attributed to fairly rare ribotype. And so there's an open question, why are only select few ribotypes making up most of the infections? What is special about these ribotypes in the first place? But also by looking at patterns over time, we can ask additional questions. So for example, I mentioned ribotype 027, which here is in yellow-orange, is the main globally dominant ribotype, and it's fairly hypervirulent. How come has it been in decline in Texas, which is also the same pattern we've been noticing across the globe, that it's been declining over the past decade? And what about in purple, we see ribotype 255, which was barely detected prior to the year 2016, but now it makes up, or as of 2018, it makes up to 5% of cases. Is there something unique about the emergence of ribotype 255? Is there a random process, or for example, could there be metabolic traits involved in the emergence of ribotype 255? So, given this epidemiology, we can ask now this broader questions I'm alluding to. How have metabolic traits contributed to the emergence of C. diff as a pathogen? And can some of these traits explain the distribution or prevalence of certain C. diff lineages or ribotypes, such as ribotype 255? So to get at that question, the Britton lab, before I joined, started profiling clinical isolates that they acquired either from Kevin Gary or from Jennifer Splindner at Texas Children, using a very basic technology called Biolog phenotype microarray plates, or PM plates. These are essentially 96 well plates that are pre-arrayed with carbon sources, a single carbon source in each well. So in each plate, you have 95 unique carbon sources. So you can take a clinical isolate, pre-culture it, then inoculate those plates and see on which wells do you see growth. And there are 2 Biolog PM plates for carbon substrates. So we have a total of 190 unique carbon substrates that we can profile. And when I first joined the lab, they already began collecting these black here. So first of all, what we're looking at is on the y-axis, log OD or log optical density. So the higher, the more growth you have, and x-axis is time. This is how you typically plot your growth curve. And this is a clinical isolate from the ribotype 27 that we have in our collection. So on minimal media, you see a classic logistic growth. exponential growth at the beginning, which in log space looks like a linear slope, and then a plateau, as you see in black. But there are sugars like in green, where you see rapid increase in OD, then a rapid decay. There are substrates like ribose and sorbitol, where you see an exponential growth followed by slower, prolonged growth over time. So this presented a unique challenge to me because I couldn't use the typical mathematical approaches for modeling these data sets. So I had to rely on something newer. And gladly, a couple of years ago, others have figured out that you can use Gaussian process regression, which is a statistical approach. I will not get into the details how it works necessarily, but allows you to model growth curves without any assumptions about their shapes. So now you can model these growth curves, and I can extract all these biologically meaningful parameters that a lot of us use from these growth curves that look unusual. So if we can get the carrying capacity, which is essentially the maximum OD that you detect, the area under the curve. We can measure the growth rate over time, which is the bottom figure that you see. For example, we can get the maximum growth rate, the maximum death rate, or even maybe something like lag time, which is the time it takes to enter exponential growth. And it's so flexible that I've even adapted it to do things like detecting and characterizing dioxic shifts. So if you're not familiar with dioxic shifts, here's an example. You have C. diff of an unknown ribotype grown on glucose, 20 millimolar. In the first 10 hours, it grows really well. So most likely what's happening is using glucose to grow to an OD or a log OD of 1.5, but it's depleting the glucose. But what's likely happening is that there's some sort of byproduct of the process that now it shifts to growing on it, on that byproduct, which gives it a bit of a boost in terms of growth, albeit at a much slower growth rate, as you see on the bottom in terms of the figure showing a growth rate over time. So I was really glad with the results of this approach, and others in the lab wanted to use it in collaborators as well. So I put it together as software, and it was published about a couple of weeks ago. So feel free to check it out. There's extensive documentation. I did my best to make it as user-friendly as possible. But if you have any questions, I'm almost happy to answer it. And it can do more than infer parameters. It can test for functional differences. It can let you visualize and do some basic quality control on your data sets. So now they know we have this sort of experimental biolog approach coupled with a computational analysis framework. We can integrate them to try to understand or get at the questions that I presented earlier. So we had profiled more than 60 isolates representing 12 ribotypes of C. diff using the biolog assays, and that I can put them into my computational framework, and then we can look at the results in a bird's eye view. For example, we can think, I'm interested in a particular ribotype like 002, which substrates support its growth? Alternatively, you can think, I'm interested in a substrate, say threonine amino acid, which ribotypes can grow on it? So using this framework, now we can start addressing very specific questions. And I'm going to run you very briefly through all these 4 questions. So first one is, what is the relative fitness of C. diff on different carbon sources? Now we do know a lot about C. diff, about the metabolism of C. diff, how it grows in different things, but a lot of that work is limited to lab adapted strains of C. diff. And seldom do we see growth assays of C. diff from clinical isolates. So this is, it presents an interesting opportunity to look at how the kind of like the pangenome of clinical isolates of C. diff and whether the nutritional preferences are the same as we expect from lab adapted strains and from what we know about C. diff in general. And in fact, that does seem to be the case. There seems to be consistent ability to grow on certain substrates. So this figure is a bit busy, so I'm going to run you through it. Basically, from the 190 unique carbon substrates available in the biolog dataset, I'm filtering out or only keeping here in this figure the substrates that consistently C. diff can grow on, or at least a select subset of C. diff strains can grow on. And then I'm ranking them by median growth. So if you look at the top, for example, that's glucose. And each isolate or each dot circle is the growth of an isolate on glucose in terms of optical density. So you have the distribution of about 60 isolates growing on glucose. You can see the median and the interquartile range. And for the most part, things grow really well on glucose, and it's a fairly tight distribution. But then we can, instead of looking at what substrate at a time, we can sort of categorize them based on The types of substrates. So on the top, you have something like simple sugars that tend to be giving allow CDF to go to high OD. Then you have sugar derivatives such as sialic acid, glucanac, but as well, then you have amino acids, which we know CDF really likes, di and trisaccharides. I mentioned previously trehalose. Then you have sugar and amino alcohols, and finally, short chain fatty acid derivatives. These sort of nutritional preferences are not necessarily surprising. Simple sugars are highly reduced, so they have a lot of energy that C. diff can extract, and they can use it to create a lot more biomass. But it does, give us a better idea about the sort of the ranking of these different nutrients in terms of the preferences of C. diff, as well as the distribution. So, for example, if you look at something like trehalose on the bottom, you see a very wide distribution in terms of how isolates grow on it. Trehalose is about the 7th from the bottom. So overall, the median is fairly low, but there's this right tail end of substrates that seem to be growing better, which gets me to the next question. Are there substrates that are variably utilized by distinct lineages? So now that we have this data set, can we predict which substrates maybe certain ribotypes are better adept at growing on, like we expect for trehalose? So going back to this figure that I just showed you, I pointed to trehalose. The average growth is fairly low, but there's this right tail end that's actually dominated by ribotypes such as 027, 078, which we know have either gene variants or gene operons that are involved in growing on trehalose at very low concentrations. But there are other substrates that you can, by looking at this, kind of make that prediction. But I'm going to only show you validation for some of those. So now we can go back to our freezer stock, look at all the isolates, take them out on the bench, and try to grow them again on known concentrations of these substrates. So for example, trehalose. Here on the y-axis is the fold change. All you need to know is that at 1, that indicates no difference in growth. Higher than 1, that means that particular isolate will grow on that sugar, for example, will grow on trehalose really well. So, and then we can rank those isolates and color code them by ribotype. And we see that on the right tail, which are the isolates that grow the best on trehalose, they're dominated by 027, ribotype 027, ribotype 078 in red, and clade 5 animal-associated ribotypes in pink, which is what we expect based on what we know from our previous work. So as a new result, I looked into a different sugar, tagatose, which some of the data essentially indicated that there might be some sort of variation. And it turns out that animal-associated ribotypes, as we see in pink, seem to like it the most. What's interesting about tagatose is it's abundant naturally in vegetables and fruits, but the food industry has yet to figure out a way to mass produce it, although there's been a lot of investment to try to figure that out. So my prediction is the next 5 to 10 years are more like there's going to be probably some sort of breakthrough that enable them to include it, because not only is it sweet enough, but it has also chemical properties similar to how trehalose does that makes it as an attractive alternative to a lot of sugar, dietary sugars. So you can imagine, you can speculate, for example, let's say tagatose becomes the most dominant sugar in our diet. Would we now expect these animal-associated ribotypes to start causing infection in humans as well? If, in fact, tagatose provides a ecological niche or a nutritional niche for C. diff. As a last example, I want to talk about a substrate that's not really common in the diet, but could be available to human other ways. So salicin, as you can see on the far left, it's a sugar linked to a phenol group. And the data indicated that this might be some variability. And when I did this validation on 30 millimolar salicin, you notice that 027 ribotype in orange and 017 ribotype in yellow seem to grow the best on it. What's interesting here is that I mentioned before, the ribotype 027 is the globally dominant lineage. Ribotype 017 is not globally dominant, but it does dominate in East Asia. It is an emergent strain in East Asia, particularly like Japan and China. And salicin is part of the group of what's called salicylates, and they're derivatives of salicylic acid. For example, aspirin is a derivative of salicylic acid. A lot of these salicylates are found in plants, and they're involved in protection from damage and stress. So while they might not be a major component of diet, they are available in plants, but they could also often be used in medicinal purposes, whether directly or indirectly. So on to the next question. Now we can focus, instead of looking in general, bird's eye view at all these different substrates, all the different habitats, we can focus on a particular habitat, let's say 255. I mentioned earlier that 255 seems to have emerged over the past 5 years or so, makes up about 5%, perhaps more, of cases in the state of Texas. And I actually found out over the past year that it's starting to show up in other states such as Minnesota and Massachusetts. So is there something interesting about them? In particular, are the metabolic traits are interesting? So when I go back to the biologic results here, I'm only showing you a sample of the results. And if you look at the top, for example, this is the results of growth on fructose. 255 is in purple. These are the different isolates tested. And the x-axis is essentially the growth, or the amount of increase in growth due to fructose. And overall, 255 does grow better on fructose than other isolates. Same thing with these other substrates that I highlight here, like glucosamine, ribose, and methionine. But what's striking is the ribose result. When you look at ribose, almost none of the other isolates grow to a decent or a modest OD. But 255 consistently does grow on ribose. And I went back using a slightly different experimental protocol, tried to validate it. So I went back to our freezer stock, pulled out a bunch of isolates, and in fact, that does seem to be the case. So if you look at no concentrations of ribose, 24 millimolar or 12 millimolar, 255 isolates tend to, on average, grow much better on ribose than other isolates, representing a variety of different ribotypes. And the same thing for fructose. So there might be two things involved here, that perhaps ribotype 255 has a slight growth advantage where it can more efficiently grow on a lot of these substrates, and that might be heightened for ribose in particular. But I won't get in, because we're limited in time, I won't get into it. It does seem to be that in a lot of cases, when things don't grow in ribose, that might have to do with with slight different experimental protocols, which I'm currently trying to figure that out. What's interesting about ribose, it's a 5-carbon sugar. It's heavily abundant. It's one of the primary components of DNA and RNA. So if you remember your basic biology, you have the phosphate sugar backbone of DNA and RNA, and that sugar is ribose. It's been granted generally regarded as safe status, which means that a lot of food industry can include it in fairly generous limit in our many different items. It's typically marketed for energy supplements in beverages and bars, but also as a substitute for typical dietary sugar. And it gets malabsorbed, probably similar to fructose, where your body can absorb it to a certain extent, but if you consume too much of it, it starts to be very severely malabsorbed, and your gut microbiome will have access to it rather than being absorbed through your body. And what's interesting that if you're a gut microbe and you can scavenge ribose or figure out a way to do it efficiently, you might have a competitive advantage in the gut. There was recent work by Eric Martin's group showing that a common gut commensal, Bacteroides have figured out a way to scavenge ribose from nucleosides and nucleotides, and that does give it in mouse model a competitive advantage under certain dietary backgrounds. So you can imagine if C. diff or any other pathogen does figure out a way to also scavenge ribose, which can be abundant in certain diets, then they might have an opportunity to emerge and dominate as a lineage. The final data figure I want to show has to do with this idea that I presented earlier, where genomic evidence suggests that human-associated strains may be more adept at growing on simple sugars than animal-associated strains of C. diff. Since we had this sort of, this framework and a lot of this data and isolates, it seems like a very interesting question that we can easily test. And so I went ahead to that using the same data set. And this is, again, the result I was alluding to from Trepalala's group, where they suggest that human-associated habitats have been selected for genes that involved in metabolism. So similar assay, but here I'm shifting the figures by 90 degrees. So on the x-axis is your fold change. So higher than 1 means higher growth. And I'm breaking down all the isolates by clades, phylogenetic clades. And that's because clade 5 includes the animal-associated ribotypes, which are in pink. Rhabita 078 in red is also animal-associated, but causes a lot of infections in human as well. So right away, you notice a counterintuitive result based on, a result that counters our hypothesis. It does seem like the animal-associated microbes, or clade 5, grows better on fructose than everything else. But that does seem to be driven by primarily the pink isolates, which are the animal-associated microbes. That is the case as well for glucose. Again, you see much better growth for clade 5 on glucose. To a certain extent, that seems to be the case for mannose, but definitely also the case for tagatose. So our original hypothesis was that human-associated strains will grow better on these simple sugars, but in fact, we see the opposite. So there's a couple of potential explanations here. This is a single experimental protocol using a single recipe of minimal media. I'm planning to try a bunch of different protocols as well. It could be that the analysis by the Trevor group was biased to certain genes that doesn't include enough animal-associated genomes or strains, or the interpretation of the results actually was biased as well. Now, it's still all ongoing work, but this gets me to kind of wrapping up. I've sort of showed you that that we kind of know the nutritional preferences of C. diff, and it seems to be fairly consistent for clinical isolates, but that there are substrates that can be variably utilized by distinct lineages. For example, I showed you tagatose and salicin. Rabitab 255 may have a metabolic advantage, and that could be more striking for ribose. And finally, human associated strains actually grow worse on simple sugar than animal-associated strains. So it's all ongoing work, and I have more exciting experiments lined up. But now I want to thank Knowledge Funding, in particular the training grant from Baylor College of Medicine in Pediatric Gastroenterology, our collaborators, Rob, and the rest of the lab. In particular, here I name people involved in C. diff in the lab. And with that, I'll take any questions.
That was great. I have a question. This is Heidi Kaplan. I was wondering, the salicylate, I was a little confused about whether or not the C. diff strains will grow on salicylate itself. I know that the sugar that you showed was some kind of a derivative of that. So could you clarify that?
Yeah, so the sugar salicin is a derivative of salicylic acid. We haven't tested yet if it grows in other salicylic acid derivatives or salicylates in general, but that seems like a potential avenue.
Okay, thank you. That was great. Really interesting stuff. Thanks.
Thank you, Heidi.
I have a question too. Do you have
yes.
do you have the genome of all of those strains?
I have genomes for a lot of those strains. The 255 ribotype strains, we have genomes from Caenorhabditis. I have for the Trevor Lawley group, for that genome analysis, I have access to a lot of those genomes, the animal associated and the human associated, and I've been actually analyzing those.
Is there any obvious differences between, that can be associated with the preference of sugar, or all those pathways are present in a similar fashion across all the clade?
I haven't done that analysis, but in collaboration, Jennifer Spillner currently has work that's, I think, still in revision, where have been doing will sequence a lot of the genomes and have done metabolic modeling, in fact, to try to see how well does the biologic data match up with the metabolic modeling predictions. So I think that should be coming out soon, but there does seem to be correlation between those two.
All right. Thank you so much, Firas. That was a really cool talk. I think that it is about time to move on to Adrien's talk. talk, and so I'll just give a quick introduction. Adrien Assie did his Ph.D. in the Max Planck Institute for Marine Biology with Dr. Nicole Duboulier, and he was studying bacterial symbionts associated with deep sea invertebrates. Now Adrien is at Baylor College of Medicine doing his postdoctoral studies with Buck Samuel, and he has established a model microbial collection of C. elegans symbionts that I think he'll be telling us a little bit about today, and how he is studying their interaction with the gut environment and diet. Take it away, Adrien.
All right, thank you for the introduction. All right, hello everyone. Today I'm going to be talking about my work on investigating host-microbe metabolic interaction using the C. elegans model. I think I don't need to really introduce this crowd what the gut microbiome is, or that the gut microbiome is an ecosystem where it's a compartment that hosts a rich community of bacteria and lives in close interaction with a host. Those interactions are in a fine balance, and when usually that balance has a positive effect for the host, it's considered healthy. But any type of little changes in that balance, like Firas just highlighted in his talk, can actually drive the microbiome towards a state of dysbiosis and disease. So in my work today, we're going to dive into intestinal host microbe interaction with, on one side, we have the host side, which is for animals and mammals. It's a collection of like compartments connected to each other's, where the interface with the gut is complex tissues of like cells with specific functions, that is protections, immunity, synthesis of mucus, and so on. And on the other side, we have the bacteria, which is a collection of like a rich collection of different species which are interacting with the environment, with themselves, and with the host. Bring all together, this is a very complex system where every organism is actually interacting with the others and can influence the whole balance in here. And even if this is a complex puzzles of arrows, like each one of these arrows is actually the sum of many different potential interaction. And here I'm highlighting in like this little white bubble that one black arrow can be the collection of like dozens to hundreds of potential metabolic interaction between two bacteria. And to illustrate how complex that can be, if we just do like a simple prediction of like which compound can be shared between different organisms, here we are looking at C. elegans in blue. The blue are the enzymatic reaction present in C. elegans. This is and in green and orange are the enzymatic reaction present in two other bacteria. And this is already looking like a very complex yarn ball connected to each other. But I'm gonna show you here today how I'm approaching that problem and how we dissect interaction. I'm gonna first start by explaining to you why C. elegans, the model nematode I'm working with, is a very cool model. Then I'm going to show you, explain a little tool we developed in the lab where we go from 16S RNA profile to functional profile. And then finally, I will dive into the role of amino acid in host microbe interaction. So why is C. elegans a cool model? Like I just explained to you, like mammals, higher organisms have like, it's a very complex system. And the main advantage of C. elegans is simplicity, where the gut is multiple compartments, complex epithelial organization, specialized cell function. In C. elegans, you have like one long compartment, which is just 10 pair of cells, which kind of all do the same thing, maybe difference with the first and the last pair, which make it very easy to tease apart like fundamental molecular interaction. In addition to that, C. elegans has a plethora of genetic tools available. I think every single knockout gene you can think of exists, and you can order it from a data bank. It's simple to raise, has a short lifespan, so you can easily do high throughput experiments. Like in the equivalent of shoebox, you can have like dozens and dozens of replicate for your experiment. In addition to that, we're working with defined microbial community, where we in the Samuel lab have 500 plus bacteria that have been all isolated from wild C. elegans. And here now, these days, we're working with like simplified community, which one of them is called the CMBio, which is a collection of 12 bacteria, which are usually found associated with C. elegans in the wild. It's simple and easy to use. Every bacteria is completely sequenced, and now we have fluorescent genomes and some mutant available from some of them. During this talk today, I'm going to talk multiple times about even the simpler community where we just use 2 different bacteria, and this is just to start to tease apart like individual interaction between the members present in our collection. Speaking of 2 bacteria, I'm going to speak over and over about 2 of our most favorite bugs. So one of them is Microbacterium. It's an alpha proteobacteria, and we really like that bacteria because it has some very clear phenotype across different host strains. In some strains of C. elegans, that's going to whatever the bacteria are going to put in the mix, they're always going to be 80 to 90% of the bacteria colonizing the gut of the worms. And in other host strain, that is only like 2 to 5%. So that's a clear phenotype where we can actually dive into and look for what caused those differences. This bacteria, it has a wide range of metabolic potential. They can pretty much eat or produce any compounds they need. On opposite, well, I'm also going to talk a lot about Myroides, which is a flavobacteria. And the main interest we have on this bacteria is that it has a negative correlation with Microbacterium. So when we have a lot of Microbacterium, we don't have a lot of Myroides. And when we have a lot of Myroides, we don't have Microbacterium. And in contrast, that bacteria needs to import a lot of things from the environment and cannot like synthesize a lot of amino acids, for example. Setting experiment with C. elegans is also very easy. Like you take the bacteria you want to work with, you mix them together, you plate them on petri dish, you let this bacteria grow for a couple of hours to overnight. Then you can use what we call synchronized nematode, where if we bleach a population of nematode, we can just get germ-free eggs that can then be raised, hatch all at the same time, and then we have like a whole population that every worm is at the same life stage. That makes it easier for experiment like microscopy work and so on later. This also gives us like a full control on the microbial community that we expose to the worms, and also the nutrient that they're exposed to by controlling the composition of the growing media. So let's dive into the experiment we did. So with the CMBio community, we did an experiment, and that relied to the diet, where we grew the CMBio community with our C. elegans worm in one media where it was rich in nutrient, which is called NGM, and that's the equivalent of your traditional microbial LB, where everything is available in excess. And in opposite, we use a media called PFM, which is peptone-free media, which is the same thing without yeast extract or peptone in the media, which is like much more restricted in nutrients. We grow our worm with our bacteria, we wait a couple of days, then we extract the DNA, we do 16S amplicon sequencing, and then we can estimate the population that grows inside the gut of our worms. And immediately you can already see that there is like stark difference between the 2 populations. That left panel here is like the community growing on the rich media, and then right panel is the community growing in poor media. And you can see that where in the rich media there is 3 different bacteria that are dominating the population, this is completely overtaken by that blue colors, which is actually the Microbacterium in nutrient limited environment. So having that 16S estimated population gives us an idea of like what's different between conditions, but doesn't tell you what's there. And because worms are very small and the bacteria density, which is colonizing that, is even smaller, it's very difficult to do any type of fancy omics experiment like transcriptomics or proteomics, where you're going to need a lot of material to just get a little fraction of the microbial portion. So for that, we decided to see if we could predict the functional profile of our community based on the genomes. We have the genomes of all the bacteria present here. And here what you can see is for each bacteria, we know the genetic profile of that bacteria. So we can then basically multiply the 16S abundance with the genome, the gene present in each bacteria, and have an estimated abundance of genes in every sample. We do that with every bacteria. And then at the end, we do have a gene community profile. What does it look like? It basically looks like this, where instead of having a table where you have 16S counts, you have a table where you have gene or pathway count. So I developed a tool called Wormbiome, which does that and use two different databases. One is the Metacyc database, which is going through the presence absence of metabolic pathways, and the other one is the KEGG database, where we're looking at the abundance of functional genes. So if we look at what's different in our experiment, what's the difference between the peptone-free media and the nutrient-rich media? Here, what we see, all the single dot is one pathway, and the color of the pathway represents its category, if it's amino acid, carbohydrate, and so on. And the size of the dot represents in how many different bacteria present in our experiment that the pathway is present. That generates a lot of possibility, a lot of rabbit hole to disappear in. But for the sake of time today, I'm just going to focus on a filtered amino acid biosynthesis. So here, that's quite interesting to look at that, because we see that the community that is colonizing a worm in nutrient-poor environment is enriched in more essential amino acids biosynthesis pathways. And why do I say essential amino acid? Is because all the amino acids that are highlighted here are actually needed by the worm to get through the nutrient. It cannot biosynthesize it, which is kind of suggesting that the worm is actually selecting maybe the bacteria that they can patch his metabolic needs. So let's dive in a bit more into that amino acid influence on host-microbe interaction. So if we look at that as with a bird's eye view, amino acid enter the gut through digested bacteria or the environmental availability. The host needs 11 amino acids that it cannot biosynthesize. Like 9 of them are actually shared with humans. Those amino acids are involved in the building blocks of proteins. They're also carbon source, as Firas highlighted, but they also involve in metabolism and molecular signaling. The host will always need to uptake them, whatever happens. And most of the amino acid transporter in C. elegans and in many human systems actually exchange a non-essential amino acid for an essential amino acid. So there is always a constant flux of amino acid in the environment, which can actually affect the composition of the bacterial community. On the other hand, bacteria have a wide spread of metabolic potential. Some bacteria can synthesize everything, and some others have a much more specialized metabolic potentials, knowing that they will always uptake it from the environment. So how do we study that? But first, I disrupt the amino acid availability in the environment. How did I do that? I took my experimental setup. I use a minimal media, which is just salt and glucose. And to that, I complemented with different mix of amino acid. I then grew on that either a SynBio community or a combination of pairwise bacteria, always with fluorescent bacteria based on that. Then we use our gem-free synchronized nematode, grow that on it, and then when they reach adulthood, we just take a picture on the microscope and measure the fluorescence. That's how it looked like. We do have a little tool called Worm Aligner, which is taking when take a microscopy picture, you take a picture of the worms, and then in export, the bright field channel and then the fluorescence channel. And then using tools like MIG and script, we can actually translate that fluorescence picture in hard data, where we extract the pixel intensity of the fluorescence for it and analyze that. Like this, for example, here, what you're seeing here, this is a picture, and this is actually a heat map of the pixel intensity, which is like showing that we actually can recover the information. In this case, I am just collapsing the total intensity and looking at the abundance per worm. So how does it look like? So this is going to, how the very complicated figure coming next is going to look like. On the x-axis, we have the green fluorescence, which is going to be related to the abundance of Acrobactrum. The more it's on the right, the more Acrobactrum is present. On the left, it's going to be a lot of red fluorescence, which is going to be a variable bacteria. And the more the more it is, the more abundant it is. Each dot in the graph is going to be one worm. And that setup, by just looking at fluorescence, allows us to actually do high throughput and process a lot of different conditions at the same time. So here, what you're seeing is the experiment ran with different amino acid mixes. So we have 6 columns where we do have all amino acid, no amino acid, and a selection of 4 different amino acids that were coming out as significant in our analysis. So no arginine, no isoleucine, no proline, and no tryptophan. Then we tested different combination of bacteria. The first row is the CMBio community, with in green still the Acrobactrum and in red the Myroides, because it's our favorite bug. Then just the Acrobactrum and Myroides, just the Acrobactrum and different other bacteria present on the CMBio. For the sake of time, we're just going to focus on 2 of those combinations, which is the CMBio and the Myroides Acrobactrum, because we're actually looking at the same abundance of bacteria. What's interesting here is like, so this is the same data as you just saw, but all the points are now overlapped. So here, why did I do that? Because here you have the clear overview that when the complete community is there, all the dots are kind of overlapping, except that light blue, which is no amino acid, indicating that the community can rescue the loss of a single amino acid in the environment, and we are going to always have the same level of colonization between Acrobactrum and Myroides. However, if we just look at a Myroides Acrobactrum combination, we see that even if we do have a lot of color overlap, we do have like this yellow and orange shift, which is in the case of no isoleucine or no tryptophan, the Myroides actually colonize about a log less the worm, showing that Acrobactrum is actually not sharing those 2 amino acids with Myroides, and then a single change of amino acid in the environment can influence how different bacteria can interact together in the gut. Now we move to the next step, is how do we test the amino acid influence on community assembly? By disrupting the host exchange. So the host is importing amino acid from the gut through different families of protein, and I'm focusing on 2 main ones, which are shared with humans, is the AAT genes and the PEP genes. So those are the 2 main ones, and to test this influence, we actually inactivate these genes by using RNAi. In C. elegans, it's actually quite easy to do. First, you grow the worm on a loan of E. coli expressing the specific RNAi for the gene you want to disrupt. Then you wash the RNAi off, and then you grow your microbial bacteria. You put the worm on the new loan of bacteria you want to test. In this case, we kept it easy and just put the worm on a loan of red Myroides and green Acrobactrum. And then same thing again, we measure the fluorescence present in every worm. And here, it actually has some differences. So what you can see here, let's focus first on the microscopy picture here, which are single worms, and this is an empty vector, which is basically just an E. coli. And you can see that the long worm, which is colonized, evenly colonized by the Acrobactrum. Then we have these 2 different AAT genes, where AAT1 is still colonized to the same amount, but A86 has almost barely any Ochrobacterium present in the gut. And if we look at the distribution of all the genes that we tested, it actually worked quite well. So that green line here is the level, the median colonization of Ochrobacterium with just E. coli without RNAi. And then the red one is just for Microides. So if we see that for A81, the inactivation of A81 didn't affect the colonization of Ochrobacterium, we can see that all the other transporter we tested actually lowered the colonization level from the bacteria, indicating that the exchange of amino acid between the host and the environment is also influencing how well bacteria can colonize the gut. And finally, that's our latest way of work, where we disrupt the metabolic potential of bacteria through knockout mutants. And that's the work from the research associated in our lab, Dana Blackburn. And we have some early promising results. So how do we do that? We're actually using T&seq libraries, where we're introducing inside the bacteria a library of transposon, which are all barcoded. This transposon, each one of these transposon is going to disrupt one single gene and is associated with one barcode. So what we have at that point is like one tube with thousands of barcoded mutants that we can use for experiments. Let's say we want to see that bacteria growing with worms, and then at the end, we just do the same thing, except this time we don't do a 16S amplicon library, we are doing a barcode amplicon library. And then we're just like trying to collect all the barcode that we can have at the end, and the barcode that are missing from our library are actually associated with genes who were important for the colonization of the worm. So at the moment, we're working on that. We do have the library ready, but we are doing performing our first experiment, but everything is still on work. But we kind of cheated a little bit, and we wanted to get some result quick, very fast. So we isolated 96 of this colony. So we instead of having like one tube with all of our barcode, we have like 96 bacteria with a single barcode, and perform the colonization experiment to see if we can have any promising results. And we do have we have like a handful of like mutants where we could observe like differences in colonization of the Ochrobacterium with two different strains of worms. And I'm just going to focus on two today, which is the first one is related to UTP and polysaccharide synthesis. It's a dehydroreutase, which is actually downstream of the biosynthesis of aspartate and glutamine. So it's kind of still related with the amino acid. And this one is very exciting because like in every combination we tried, we observe actually a lower colonization of the Ochrobacterium, indicating that amino acid related pathway is important for colonization. The other gene of interest is a sarcosine oxidase, which is downstream of the glycine betaine biosynthesis pathways. And this one is also interesting because it lowered the colonization in only with one of our strain, where like we can see that like bacteria colonize like less well in that one, whereas in our main host strain there is no changes. So that's promising results showing that like bacteria and amino acid is also the metabolic potential of bacteria is related to their ability of colonizing. So in summary, I hope I showed you today that amino acids are important at every level of the host microbe interaction, where the amino acid availability in the environment is going to influence pairwise microbial association, but different community composition are able to resist those changes and rescue single amino acid deficiency. Amino acid import-export from the host also influence how well some bacteria can colonize, suggesting that the host is probably kind of shaping his own microbiota by controlling which nutrient is available for the bacteria to grow. And finally, the amino acids are still important at the bacterial level, where they need those specific pathways to colonize the gut. The future work is to get our T&seq going to basically and explore every single, to continue the work of every single point I raised today, where we could track the amino acid interaction between organisms. So here I just show you like 4 amino acid dependency and only a handful of combination. But now that we have the setup ready, we can actually expand that and do like a complete map of like which amino acid can be shared between which bacteria. And then ultimately, I want to understand the flux of amino acid happening in the gut microbiome and how you go from the left healthy microbiome to a sick one on the right. With that, I would thank you for attention. Also, I thank you all the people in my lab, especially Dana Blackburn, that does a lot of the TNSIC work and the bacterial work, and Daniela Vidal, who did some of the early mutant work for the fluorescent bacteria. Our collaborator, then the Dutch Bauer, who created the magic pools, which is how you can create easy TNSIC library to do our work. And with that, I'm happy to take any questions.
I can start. So when you were looking at the effect of the amino acid transporter mutants on microbiome composition, do you have any hypothesis or hint of whether it is due to the host controlling nutrient availability for the bacteria, or do you think that it's the host senses that it's lacking certain amino acids and inducing some kind of more complex, like almost immune response?
I think it's a mix of both. I think that there is like the essential need from the host to uptake the amino acid it cannot biosynthesize, and then it kind of influence which bacteria can colonize, because like by taking off certain amino acid and putting some amino acid in, you do have like an influence of the chemical environment, like of the guts. And then on an evolutionary standpoint, it's probably like just a selection of like which bacteria is the best combination, right? Like some bacteria are going to abuse that system and probably be deleterious for the whole network balance, and those worms are going to die and not pass that exchange to the offsprings. Whereas like the one that is going to select the bacteria that are going to like it and enhance the exchange rate are going to continue to. It's a bit confusing answer, I'm sorry, but I think, yeah, sorry.
Adrien, this is Heidi Kaplan. I was intrigued and not aware of the fact that, so you were saying that when an organism takes in one amino acid, it exchanges it for another amino acid. I've never seen that before. Is that because I'm a bacterial person? Is this something that the worms do? And it seems as if that's the case, that you would have it. I just don't understand that. Is that like, it seems like you'd be bringing in one amino acid from, as you're saying, suggesting the bacteria are producing, but then you'd be putting out another amino acid. So that seems kind of, I don't know. I just haven't heard about that before.
Okay, so for the sake of the story, I'm just highlighting those, but a lot of the amino acid are transporter are classical ones. So you're going to have like ABC transporters, which are going to be ATP fueled, and other ones which are going to be just exchanging ions. But some of the so like that's like some of the free amino acid transporters are part of the H80 family transporters, and I've read that like they're actually exchanging it's amino acid exchange transporters. That fits very well with my narrative at the moment. I have yet to test it in our system. Like one of the experiments I'm actually planning to do is actually just like, because now we've seen that we can silence the transporters, is actually to do the same experiment and just measure the content, the amino acid content of the worms afterwards with like mass spectrometry to just like see if the inactivation of amino acid transporter is actually increasing the abundance of amino acid in the worm. That would kind of like suggest which amino acid is actually exchanged with the environment.
Yeah, it just seems kind of very complex, and I'm just sort of wondering, does it bring it in and then metabolize it and then send it out as another? Because otherwise, like what would happen if you didn't have the one inside that was required for the exchange, then you might not be able to bring in this amino acid if you didn't have the one to export. I don't know. Anyway, it's interesting concept. So thanks. I won't take up any more time with that question, but thank you.
Any other questions? If anybody would like, they can type a question into the chat and I can read it. All right, well, thank you so much, Adrien and Firas. This was a really great session. Cool to hear about diet and the microbiome in a few different systems. And so I'd like to remind everybody that we have MicroTalks on the last Wednesday of every month at 4 p.m. And we look forward to seeing you next month.
Thank you.